Bismillahirrahmanirrahim. We start the chapter on the last chapter, which is inheritance, and this is 5090 O level biology. And this is the first video on this chapter, and hopefully, we should be able to complete this chapter in a day or two. Now, the first point in the syllabus is describe the difference between continuous and discontinuous variation and give examples of each. So, now continuous variation, as you can see here, the example is height of humans. Now on the y-axis you have the number of people and here you have the height. So continuous variation means that there are many intermediates. There are many intermediates. You can't have people heights, now of course we will be talking about height of 16-year-old boys or 16-year-old girls. So that height is going to be very variable. And it's going to be very different in Pakistan, maybe it's very different in China, maybe it's very different in Japan. So continuous variation means many intermediates, and please remember continuous variation is affected by the environment. Like for instance, I have the genetic predisposition to be 5 feet 6, but then I didn't get enough food in my diet, I didn't get enough proteins, I suffer from protein malnutrition. Well, I'm not going to grow that tall. Similarly, a seed which is planted should grow 3 feet tall. The bush should be 3 feet tall. But then, of course, it didn't get enough nitrates, it didn't get enough light, so it did not grow tall. So continuous variation is affected by the environment. Then let's look at an example of discontinuous variation, and that is human blood groups. So there are a few distinct categories. Like for instance, if you look at blood groups, there are only 4 blood groups: A, B, AB and O. So there are, in all the human beings anywhere in the world, there are 4 distinct categories. So you draw it as a bar chart. So the number of people or the percentage of people who have this blood group. Now this is of course going to be very variable. It's maybe going to be different in a certain city, in a certain country. So discontinuous variation. Another factor, discontinuous variation is not going to be affected by an environment. Not affected by the environment. Why is it not affected by the environment? Because it's like blood group. Now your blood group is not going to change if you are deprived of food. Your blood group is always going to remain the same. So blood group is 4 distinct categories. So discontinuous variation. Another example of discontinuous variation is gender. Either you are a male or a female, except for the few abnormalities, but we don't talk about those. Now let's look at another table here, which is a very good table explaining all this to you. Continuous variation, no distinct categories, no limit on the value, tends to be quantitative. Discontinuous variation is distinct categories, no in-between categories, and tends to be qualitative. Examples: height, weight, heart rate, finger length, leaf length. In discontinuous variation, we have tongue roller, fingerprints, eye color, blood group. Now this is represented by a line graph, this is represented by a bar diagram or a bar graph. Controlled by a lot of gene and environment. Range of phenotypes between 2 extremes. Example, height in humans. Height in humans of 16-year-old girls, or height of humans in 16-year-old boys. Now discontinuous variation is a few genes, a limited number of phenotypes with no intermediates. Example, AB and O blood groups in humans. So this is the first part of the syllabus which we've just undertook. Continuous and discontinuous variation. Next we have to study that what is a chromosome. Now a chromosome, first of all we have to understand chromosomes, genes and DNA. Now they're found in the nucleus of the cell, and the chromosome is made up of DNA, supercoiled DNA. Now this is the DNA that we have to understand. And it's a very long, so it's supercoiled into chromosomes in order to fit. And there are 46 chromosomes, but please remember these are 23 pairs in a typical human cell. And when DNA is not being used, it is tightly coiled and it's around these proteins which are called histones. And it's wrapped around these little ping pong balls which are called histones, and this DNA is like a ladder, and it's a double helix. And is wrapped around these proteins, and this is what is a chromosome. And of course, this is a chromosome. It's got a chromatid attached to it. So this is one chromosome, and this is when DNA replication has taken place and a copy has been formed. Now chromosomes exist in two different states. Now this is the first state, just one chromosome. Now before DNA replication, chromosomes have one chromatid. This is the one. After DNA replication, now they have two sister chromatids held together at this point, which is called the centromere. And each chromatid is one piece of DNA with its supporting proteins. Why is DNA replication necessary? Because you see, what you remember from mitosis, the 46 chromosomes have to become 92, because then this is going to divide into two cells with 46 in each. So DNA replication has to take place before the cell can divide into two. Then the next point to be understood is state that DNA is divided up into sections called genes. So genes is a portion of the chromosome. So this is the one gene, this is another gene, this is another gene. So gene 1, gene 2. So what you need to understand is that genes are portions of DNA which code for a characteristic. Or we can say they code for, the characteristic is coded by a protein. So basically genes code for proteins, and one gene codes for one specific protein. You can't say this is a gene for enzymes. You have to be specific. This is a gene for pepsin. So you can't use a broad category like enzymes and say this is a gene for enzymes. No, there can be no such thing. It has to be specific. This is the gene for pepsin. This is the gene for amylase. This is the gene for insulin. Now, as compared to that, what is allele? Allele is different versions of a gene. Like in blood group, we have 3 alleles. So different versions of a gene. Like for instance, I'm holding 4 markers. So there are 4 different colored markers that I have in my hand. Now these different forms are what cause some differences between individuals. And so all the different genes and alleles that a species has is called, it's a gene, this whole genome or the gene profile. So this is the genetic information which would be of course present in the entire 46 chromosomes of the 23 pairs. Now, a pair of homologous chromosomes means a pair of chromosomes. Homologous means that they carry the same characteristics. Like I said, 23 pairs of chromosomes. So each pair is a homologous chromosome, and one is inherited from the male parent and the other is inherited from the female parent. Then a locus is the position. A genetic locus is the location of a particular gene on a chromosome. So this would be, the locus would be the location. So that's a very simple word, locus, location. So the position. And at each genetic locus, an individual has 2 alleles, one on each of the homologous chromosomes. Now if they are both capital letters, then it is called homozygous dominant. Like I said, big B, or if it is big A, or if it is big D. These letters, of course, the Institute of Biology decides to give letters to the different characteristics. Then if it is both small letters, small b, now this is called homozygous recessive. Homo means same, homozygous recessive. But if one is capital, like big B, small b, then it is called heterozygous. So heterozygous cannot be dominant or recessive, it's just heterozygous. Heterozygous means there's one capital letter and one small letter. The capital letter shows the dominant trait. The small letter shows the recessive trait. So if we say this organism is big B, small b, it means the big B is showing its effect and the small b is not being really considered. So this is a heterozygous characteristic. And I'll give you some examples and we'll try to understand this a little more now. Then let's look at the definitions. Now, what is a dominant allele and what is a recessive allele? A dominant allele is one which will show its effect even though it is homozygous dominant or if it is heterozygous. So allele that has the same effect on the phenotype, whether it is paired with the same allele or a different one, dominant alleles are always expressed on the phenotype. An allele that has the same effect on the phenotype in a heterozygous individual, where it is combined with a recessive allele, as in a homozygous individual where they are both the same. So dominant means dominates, shows its effect. Recessive allele will only show its effect if it's in the homozygous state. An allele that has an effect on the phenotype in homozygous individuals when there are two copies of the recessive allele, only then it will show its effect. And in a heterozygous individual, this is not going to show its effect. It is hidden by the effect of the dominant allele. Then another definition is genotype and phenotype. I say very easy to remember, phenotype is the physical features. Physical features means what you can see. So you can see this is a purple flower or a white flower. So if I say purple, purple means this would be a phenotype, physical features. So the phenotypes will be purple, purple, purple and white. The genotype is the genetic makeup. And the genetic makeup is shown by letters. So we can say big P, big P. Now these letters of course are decided by the Institute of Biology, and of course this would be given in the question. So you would not be asked to make it up. You would probably be given in the question what letters to use. So if you say P, P, big P, that means it is homozygous dominant. If you say big P, small p, then it's homozygous, it's heterozygous. And if you say small p, then this is homozygous recessive. But genetic, if I ask you for the genotype, you're going to give me these letters. You're going to give me this. You're not going to say purple flower or white flower. That is if I ask you the phenotype, only then you give me that. So if I ask you the genotype, then you would give me the letters. Whether it's W, the letters A or B or D or whatever, that is what you're going to give me. So genotype is always expressed in the letters, and phenotype is of course the physical feature, and we're just going to do an example and see how you understand this. Now if I say in a question, black rabbit is crossed with a white rabbit and all the offsprings are black. Now this gives you the clue. Black is dominant. But if I had said black is crossed with white and all the offsprings are white, then it would mean white is dominant. So when you read the question, you try to understand it. You say black crossed with white rabbit and all of the offsprings are black, so black is dominant. But if I said the other way around, if I said black into white and all white, then white is dominant. So this is how we need to understand this. So now if I say black into white, all black, now and I tell you, okay, use the letter big B. So you will know this will be big B, and the white rabbits are going to be homozygous recessive. So all the offsprings are going to be big B, small b. Now this is called the F1 generation. So what is an F1 generation? Is when you cross a homozygous dominant with a homozygous recessive and all the offsprings are heterozygous. Again, when you cross a homozygous dominant with a homozygous recessive and all the offsprings are heterozygous. Now this is called the F1 generation. Now let's cross these offsprings. Now when we cross the F1 generation with each other, so this is heterozygous and you cross it with another heterozygous, then you write the gametes, you just write them separately and you circle them. And then you look at the possible combinations. This is called the probabilities. So if this big B and this small b this big B fertilizes, so you get big B. Now this would be a black rabbit. But if this fertilizes, then we get also a black rabbit. If this fertilizes, even then we get a black rabbit. But then if this is the situation, then you get a white rabbit. So the ratio here is 3:1 because these three are all black. and one is white. Now this generation is called the F2 generation. This generation, the 3:1 generation, is called the F2 generation. So F1 generation, in which we crossed a homozygous dominant with a homozygous recessive and all the offsprings were heterozygous. Then in the F2 generation we cross these heterozygous and we get a ratio of 3:1. And this of course would be, the 3:1 would be in the F2 generation. Now another possible combination is when you cross a heterozygous with a homozygous recessive. So a black rabbit is crossed with a white rabbit, and look at the possibilities. Now here we have, this is black, and these two are white. So the ratio is 2:2, which we can of course simplify and say 1:1. So when do you get a 1:1 is when you cross a heterozygous with a homozygous recessive. Now what is a test cross? A test cross is when we have a mystery black rabbit. Now we know black is a dominant trait, so we know that either they can be big B, or they can be big B, small b. But we want to know, what is this rabbit? Is it big B, is it homozygous dominant, or is it heterozygous? So we do a very easy phenomena. We cross this with a known homozygous recessive. We know that the white rabbit is recessive. So we cross it with a known homozygous recessive. So this black rabbit, big B is crossed with a white rabbit. And if this is big B, then all the kids are going to be heterozygous. So they're all going to be black. So this is going to tell us what is the genotype of the black rabbit. But if it was this, and we cross it with a known recessive, now some of the offsprings will be black and some of the offsprings will be white. So if even there is one white offspring, we know that this is a heterozygous black rabbit. So a test cross is actually to find out what is the genotype of this black rabbit, and we know black is a dominant trait. So you cross it with a known recessive. We should know what is the known recessive trait then. So if it was homozygous dominant, then of course all the offspring would be black and they would all be heterozygous. But if it was heterozygous and you crossed it with a known recessive, then some of these would be black and some of these would be white. So this is called a test cross. Now, how do we really actually handle genetics questions? Genetic questions are actually marked per line. Now, the first line is, this is parental phenotype. Parental phenotype. Now, parental phenotype, here I've written blood group A, and this is one parent is blood group A and the other parent is blood group B. Now, the second heading is parental genotype. Now, here of course I'm choosing to use this as an IA and IO. And in the B person I'm using IBIO. So IAIO and IBIO, I purposely chosen that for a reason. And then of course the third line is gametes. Now the gametes, all you have to do is just write these separately. Gametes means that this is the male, then the sperms will either contain the IA or the IO. So you just write it separately and you just circle it. You just write it with a little distance and you circle it. And in this situation also, you write IB and you circle it, and you write IO and you circle it. Now the fourth line is called offspring genotype. Now you don't need to know these headings, but you just need to do the questions under these headings. So now if this sperm fertilizes this ovum, then the offspring will have the genotype IAIB, and this will be blood group AB. But if this sperm fertilizes this ovum, these are all probabilities. So this will be IAIO. But if this one fertilizes, then we have IBIO. If this and this fertilizes, then of course we have IOIO. Now this person will be blood group A, this person will be blood group B, this person will be blood group O. So this couple has a 25% chance of having any blood group. The child can be any blood group. And this last line is called offspring phenotype. Now these are the 5 lines in doing any genetics questions. First is parental phenotype, so blood group A and blood group B, that's what we can check. We can do a test and see what blood group they are. Parental genotype is again letters, so IAIO, IBIO. Then gametes, you just write them separately and you circle them. And then you of course see the probabilities. So if you number this 1, 2, 3 and 4, the way you do it is you do 1, 3, 1, 4, then you do 2, 3 and then you do 2, 4. So 1, 3, IAIB, then 1, 4, then 2, 3, then 2 and 4. So now this couple has a probability of having any 4 of the blood groups. But okay, if they have 4 children, what will their blood groups be? Now they can be anything, because why? There's a reason why. There's a syllabus point there for you. Why observed ratios often differ from expected ratios, especially when there are small numbers of progeny. You see, if this couple had 100 children, then probably this would be the probability. But if they have only 4 children, but they could be anything. They could be 2AB, 1A, 1B, 1O, or all 4 of them different blood groups. And I give you a very simple example. I say you have a 25% chance. You have a 25% chance of getting 100 out of 100 in your maths exam. Quite difficult. But I let you only take the exam 4 times. Well, you might get it, you might not get it. But if you take the exam 100 times, then 25 times you're going to get 100 out of 100. But I didn't let you take the exam 100 times. I only let you take the exam 4 times or 3 times or maybe 5 times. So this does not work when we are talking of a small numbers of progeny. This is a syllabus point. Explain why observed ratios often differ from expected ratios, especially when there are small numbers of progeny. So in this situation, in which a couple has 4 children or 2 children, but they could be any blood group, which is just a 25% probability. If they had, of course, yes, 100 children, then the probability would be this. But if they only have 2 children or 3 children or 4 children, then it's not going to work. This probability issue is not going to work. Now, I should have done this earlier, but I hope you can understand it now. Blood group gene. Now, blood group has 3 alleles. IA1, IB2 and 3 is IO. Now, IO is recessive, of course. Now, the 4 different blood groups, for blood groups A, B, there's only one possibility. So, this will be the genotype, IAIB. And this is a phenomena of codominance. Why codominance? Because both of them are showing their effect. Like if you cross red flowers with white flowers, then all the offsprings are pink. So the red shade and the white shade are both showing its effect and all the offsprings are pink. So this is a phenomena of codominance. Codominance means both are showing its effect. Then the O group is IO. So there is only one genotype for AB and there is only one genotype for O group. Then the A group people, there are two possible genotypes. They can be IA, this is homozygous dominant, or they can be heterozygous IA and IO because IO is recessive. The B group people can be IB, and they can be IBIO. So what do we have? We have 4 blood groups, 1, 2, 3, 4, so 4 phenotypes, but we have 6 genotypes, number 1, number 2, number 3, number 4, number 5 and number 6. So there are 6 possible genotypes, and that's a very favorite MCQ. So the blood group alleles are 3. We have 4 phenotypes, 4 different types of blood groups, but we have 6 different types of genotypes. 1, 2, 3, 4, 5 and 6 different genotypes. So this is the alleles, and we all have the blood group gene, but we have different alleles. I mean, if you're O group, well then you're IO. If you are group AB, then you are IAIB. We all have the gene, but we have different alleles. Alleles is the alternative form of a gene. Alternative form of a gene. This finishes the video 1, which will then continue to the second video, which will continue with the rest of this chapter. Thank you for now.